video is going to get you through the microbial metabolism notes, which are in Chapter 6. You should have already gone over metabolism in a little bit of detail in whatever freshman bio or AMP course that you've taken. We're going to go into this in a little bit more detail, and we're going to talk about some of the differences between prokaryotic metabolism and eukaryotic metabolism as well. As always, we start with an important person about relating to metabolism, and so we come back to Louis Pasteur. We talked about him before when we were talking about spontaneous generation. He helped to disprove the idea of spontaneous generation as related to microbes. For the metabolism part, what he was curious about was how can you turn grape juice into wine? And they knew at the time that you had to have organisms in order to carry that process out. Essentially what he noticed was that as the yeast within the grape juice started to grow, remember yeast is a microorganism, but it's not a bacteria nor is it prokaryotic. Yeast is a eukaryotic fungi. It's a single-celled organism. But as it was growing in the grape juice, the level of sugar within the grape juice would lower or drop, but the amount of alcohol would increase. And so his conclusion was that the yeast was able to do something with the sugar and essentially convert it into an alcohol. Now, another gentleman, Buchner, Eduard Buchner, he showed that you don't even need live yeast in order to carry out that process. So you could crush the yeast and kill it, and there was still something within that crushed yeast that could still convert the sugar into alcohol or wine. So you don't need the living organism, you just need something in that living organism. Well, today we know that it's the enzymes present in that yeast that was capable of doing that conversion. It's actually a type of fermentation that could convert sugar into alcohol. But these two gentlemen helped us get that process started. So Pasteur showed that you need the living organism, and Buchner showed it doesn't even have to be alive. You just need something that had been in that living thing in order to carry it out. So, mentioned that word enzymes just a second ago. One of the things that I dislike about this chapter in your textbook is they kind of keep coming back to the same concept, and each time they come back to it, they talk about it in a little bit more detail. So we're going to keep coming back to enzymes multiple times. I kind of wish they just talk about it all at once, but they don't. So what are enzymes? They are usually going to be made out of protein, so they're made out of amino acids. What they do is they catalyze reactions. If you don't know what that word catalyze means, it means they speed them up. Enzymes are specific, so they can generally do one thing. They can do that one thing really well, and they can do it over and over again. That's called reusability. And so enzymes are going to help us carry out our reactions a lot faster. Bacteria have their enzymes, we have ours. There's a lot of overlap. There are some that we have and they have. We've already mentioned catalase. We are catalase positive, and so are a lot of bacterial species. One little side note here, anytime you see something that ends in ASE, like catalase ends in ASE, that almost always means it's going to be an enzyme. So lactase is an enzyme that breaks down lactose. We have oxidase enzymes, reductase enzymes, phosphatase, kinase. Anytime it ends in ASE, that's going to be cluing you in that it's an enzyme that's going to be helping to speed up some kind of reaction in the cell. Metabolism is all the reactions in a cell, not just the ones that break things down, but also the reactions that build things up. Metabolic reactions tend to be categorized into two groups. We have catabolic and anabolic reactions. Catabolic reactions tend to tear things down, so they take big polymers and then break them down into monomers, or at least smaller molecules. Anabolic reactions are the opposite. They tend to build things up, so they take monomers and build them up to make polymers. As a general rule, the catabolic reactions are going to release energy, while the anabolic reactions are going to be requiring an input of energy. Biosynthetic reactions tend to be anabolic reactions, but what biosynthetic means is we're making biological molecules like proteins or nucleic acids, something along those lines. Okay, one of the other things that's going to be very important for metabolism is ATP. We've talked about it a little bit before. It's adenosine triphosphate. It's an energy carrier present in all cells. Prokaryotic cells and eukaryotic cells all use the same ATP. This is another one that we're going to keep coming back to over and over again, but your basic structure for ATP is we get a ribose sugar, we get an adenine. Adenine plus ribose makes adenosine. There's the A part of ATP. Then we have 3 phosphates coming off the side. That's triphosphate, so that's adenosine triphosphate. These phosphates are held on with high energy bonds, so if we break those bonds, we can release the energy that had been stored in those bonds. And so anytime you want to spend an ATP, we're just going to break a phosphate group off of it. What are the subunits for macromolecules? So amino acids are used to make proteins. Nucleotides are used to make nucleic acids. Your book mentions lipids here. Lipids don't have monomers that make them, so it is a macromolecule. They just aren't made out of monomers. And so usually we're going to talk about stuff like steroids, phospholipids, triglycerides. Those are all examples of lipids, but those aren't monomers. Also, I give you, for number 4, the book doesn't mention it, but monosaccharides get used to make polysaccharides or complex sugars. And so monosaccharides are your simple sugars like glucose, fructose, ribose, deoxyribose, any of those guys in there. Next up, energy, capacity to do work, or the ability to put matter into motion, whichever way you want to say that. 2 categories of energy, potential and kinetic. Potential energy is stored energy. Kinetic energy is energy in motion. Compare and contrast photosynthetic organisms with chemoorganotrophs. So, I'm just going to skip right past that. Photosynthetic organisms, those are the ones that are capable of using light to help power some of the reactions that are present inside of the cell. Generally speaking, our photosynthetic organisms, when we talk about them, they're going to be the photoautotrophs, and so they're going to be capable of taking carbon dioxide and then reducing that to make a carbohydrate like a glucose. Really, they make G3P, but then a couple of those get put together to make glucose. For photosynthesis still, they use the kinetic energy of photons to convert it into the potential energy of those carbohydrates, and so that's why they're doing that. For the chemoorganotrophs, they have to use organic molecules to power all of their reactions, versus, again, for those photosynthesizers, they were using carbon dioxide, which is an inorganic molecule. So usually what I talk about here are just some examples of photosynthetic organisms and some examples of chemoorganotrophs. And so in terms of plants, they're of course photosynthetic, but remember, cyanobacteria, they work just like photosynthetic plants. They have the same chlorophyll, they carry out the same reactions, and so photosynthetic bacteria do exist as well. For your chemoorganotrophs, yeah, I'm showing you a dove here, but there's a bunch of bacteria that are going to work like chemoorganotrophs. That would be like E. coli, salmonella, pretty much all your pathogens are going to go into that group. Okay. A little recap on some stuff you should have had in a previous course. Whenever you see a chemical reaction, the reactants are the ones that go before the arrow. The products are the things that go after the arrow. Both reactants and products have energy, but they don't have the same amount of energy. As we talk about this chapter, we're going to primarily be talking about glucose metabolism. So what I wanted to show you, and I haven't given you the equation yet, but what we're going to be doing is essentially burning glucose to make ATP, and in the process we produce carbon dioxide and water. Well, this is the amount of energy that's present in a glucose molecule. This is the amount of energy that's present in the carbon dioxide and water that gets produced, which means all of this energy difference between the glucose and the carbon dioxide and water, that's the energy that we can use to make ATP. That's why we burn glucose, is to release all of this energy. Photosynthesizers, on the other hand, they go in the exact opposite direction. They take carbon dioxide and water and help make glucose out of that. And so all of this energy is the energy required in order for them to carry out photosynthesis. And that energy is what's coming from those photons of light that we talked about earlier. Now, one of the other things that's going to be relevant here is it would be very efficient if your cell could just split glucose directly into carbon dioxide. But if that happened, all of that energy would be released at one time. That's essentially a tiny little cellular explosion, and that is not a good thing. So one of the reasons why glucose metabolism is so complicated is because we can't do this in one step. We have to do it in a lot of little steps to release the energy a little bit at a time, so we don't damage the cell in the process of releasing all of that energy. I will mention for the majors, it's a good idea to kind of talk about Gibbs free energy in here, but for the non-majors, I don't get into that. So if you don't understand Gibbs free energy majors, you might go back to, I think it's in chapter 2 is when they talk about it in your textbook, but that'd be a good place for you to understand how that energy relates to the products and the reactants. Okay. Exergonic reactions are reactions that are going to release energy. Again, as a general rule, catabolic reactions are going to be exergonic. Endergonic reactions are reactions that require an input of energy. As a general rule, anabolic reactions are going to be endergonic reactions. Now, I mentioned glucose metabolism is really complicated. We can't just split the glucose to make the carbon dioxide all in one thing. So we're going to have all of these little sub-steps that can happen here. When it comes to carbohydrate metabolism, we have glycolysis, we have a transition step, we have the Krebs cycle, we have all of these little bitty steps that we do in order to get as much energy out of the glucose as we can, a little bit at a time. So all of those steps are what make up the metabolic pathway. The way your book says that is it's the sequence of reactions that converts a starting compound, in this case glucose, to an end product, which is going to be the carbon dioxide, the water, and then the ATP. Once again, we're going to come back and talk about enzymes in a little bit more detail. I say explain how they work. What they do is they lower the activation energy required to start a reaction. So what I usually talk about here when I'm doing this in a lecture face-to-face is we talk about the process of starting a fire. Now, a fire is an exergonic reaction. It's essentially like glucose metabolism. We're taking the carbohydrates and the other molecules that are present in whatever we're burning, and we're harvesting the energy out of it and releasing a whole bunch of heat. That happens in your cells too. So the wood that we're burning has this amount of energy. The end products, the smoke, the carbon dioxide, the water, that has this much energy. So the fire releases all of this energy that is between the reactants and then the products off down here. However, fires don't just spontaneously start. You have to put energy into that reactant in order to get that process started. So if you are trying to start a fire by rubbing two sticks together, you're going to be sitting there rubbing two sticks together for at least 30 minutes, more like a couple of hours for the average person in order to get that fire started. But the end result is still exactly the same. Now, the way an enzyme works is it lowers all that energy you're having to put into it in order to get that fire started. So that would be like having a lighter. Instead of you rubbing two sticks together for a couple of hours, you just flick the little rotary thing and you get a fire started a lot faster. So the energy required to start a reaction is called activation energy. Enzymes lower it, so they make it easier for the reaction to go ahead and get started. It's important to know what a substrate is. The substrate is the chemical that the enzyme is going to act on. For example, catalase acts on hydrogen peroxide, so catalase's substrate is hydrogen peroxide. How activation energy affects reactions. Again, that's that energy required to start the reaction. The lower it is, the faster that reaction can start. And it's very important that you understand that enzymes are specific. So catalase can only work on hydrogen peroxide. It can't do anything to alcohol. It can't do anything to bleach. Sometimes some enzymes will have a little bit of give, and so they can work on multiple types of a similar thing. So they might be able to work on a couple of different kinds of sugar that have a similar shape. But most of the time they can really just do one thing, and they do that one thing really well. Now, glycolysis is the first set of reactions that happens with glucose metabolism, and generally it's going to take a different enzyme for us to do every single one of these reactions. And so hexokinase helps us convert glucose to glucose 6-phosphate. You need a different enzyme to do the next reaction, a different enzyme to do the next one. I didn't have to memorize the enzymes necessary for this when I was taking microbiology, so I'm not going to make you memorize them either. However, I do need you to know that it takes a different enzyme to do each one of these steps, and if any one of those enzymes is missing, that means there is a halt in this process, because that next step is going to take forever in order for it to carry out, because that's how enzymes work. They speed it up. Okay, what is ATP? Again, this time I wanted to be a little bit more specific and get those components in there. So it's adenosine triphosphate, which is an adenine and a ribose to make the adenosine. And then it's those 3 phosphates coming off the side. And once again, it's just the energy carrier for a cell. Okay, didn't have another slide for the next one. When an ATP is spent, what products are released? So I mentioned earlier, you've got these high energy bonds that hold the phosphates together. When you spend an ATP, you break one of those bonds, and so what happens is an inorganic phosphate, which is what that little PI stands for, that gets split off. So that PI is that phosphate group right there, and then you have an ADP. So when you spend an ATP, you get an ADP, which is adenosine diphosphate, plus an inorganic phosphate, and then all the energy that got released, you can use that energy to do something else inside the cell. Contrast substrate level phosphorylation and oxidative phosphorylation. So substrate level, what happens there is the energy released in one exergonic reaction powers phosphorylation of ATP. Here's what I mean by that. Okay. So this is one of our steps in glycolysis. We take phosphoenol pyruvate, which is PEP, and then we convert it into pyruvate. PEP has a phosphate group to it. The enzyme comes in and it splits off that phosphate, that releases energy. That energy is then going to help take that phosphate and put it onto ADP to make ATP. So this one reaction not only converts PEP to pyruvate, it also glues a phosphate onto ADP to make ATP. So saying that again, the energy released from one exergonic reaction, this reaction, powers phosphorylation of ADP to make ATP. Let's see, oxidative phosphorylation works entirely differently. Now, in a previous chapter, we had talked about proton motive force, and I told you it was going to come back. This is the chapter that comes back to us. That energy that was generated in proton motive force powers phosphorylation. So instead of this one reaction making an ATP for us, that whole proton motive force aspect powers the creation of ADP into ATP. All right. Oh, I think I have a hyperlink here is what that says. It's not on that slide. Okay, it's on this slide. So I'm going to take you out to a video that might help explain that process a little bit better. Okay, they're just going to play groovy music here. Okay, so in our cells, oxidative phosphorylation happens in the mitochondria. In prokaryotic cell, this happens at the cytoplasmic membrane. So there is a difference in where it happens between those two cells, but aside from that, it's the same basic jazz. So glycolysis and the citric acid cycle, those happen in the cytoplasm. The electron transport chain, that happens in a membrane, again in our cells, in the mitochondria, but in prokaryotes within the cytoplasmic membrane. This little part is just for the mitochondria specifically. For the prokaryotic cell, they're just pumping it across their one membrane. Okay. So this would be the cytoplasmic membrane in a bacterial cell. We have all of these little proteins. These proteins are going to be helping to create the proton motive force that we have talked about. Now, the proteins that they're listing are specific for eukaryotic cells. We'll talk about the ones that get used in prokaryotic cells later on when we start talking about this in a little bit more detail. So you don't have to worry about these names because those aren't specific to bacteria. But we have these proteins. These proteins are going to be accepting electrons from energy carriers like NADH and FADH2. And then it's going to pass those electrons from one protein to the next. And as they do that passing, we're going to pump hydrogen ions out of the cell in the case of bacteria. And so NADH is an electron carrier used in all cells. It brings the electrons to NADH dehydrogenase. The electron is going to get passed from that to something called ubiquinone. As we pass those electrons to ubiquinone, we're also going to pump a hydrogen ion out of the cell when you're talking about prokaryotic cells. So passing those electrons, that's what allows us to pump the hydrogen ions out of the cell. And again, there's some name changes between our cells and bacterial cells, so don't focus on what they're saying in the names because that's specific for us. Now, those electrons are going to get passed to another little guy. As we pass those electrons, we can pump even more hydrogen ions out of the cell. What we're doing here is creating, again, that proton motive force, because a hydrogen ion is just a proton. We're going to end up with a bunch of hydrogen ions out of the cell and very few in the cell. So we've created a diffusion gradient, a chemical gradient. Those protons or hydrogen ions are eventually going to be cycling through a turbine that's not present in this picture just yet, called ATP synthase. And as they flow through that turbine, it spins and it uses the energy of its spinning to create ATP. So this is oxidative phosphorylation. It's a lot more complicated than one reaction powers the creation of one ATP, but this is how your cells work. Your cells are complicated. So instead of just one reaction doing it, we have to pump electrons to pump protons. The protons will flow through the turbine to help us create a bunch of ATP. I'm going to stop that video because they are longer than I need them to be right now. Okay. So, the next thing I do is I kind of recap electronegativity because this is what influences where electrons are going to go. Electrons always want to travel towards a chemical or an element that has a higher electronegativity, and so this is to show you that electronegativity trend. The element with the highest electronegativity is fluorine. Oxygen also has very high electronegativity. One of the reasons that not just you, but a lot of bacteria need oxygen is because of that high electronegativity. What it does is it has the ability to pull the electrons from that electron transport chain out so that we can keep doing the electron transport chain. So it's what's known as a terminal electron acceptor, and that's going to come back to us in just a second. Any aerobic organism, the reason why they need oxygen is because of this high electronegativity, because it can accept those electrons from another molecule. Energy source is the molecule that's going to be providing us with energy, essentially, but it's very often going to be in the form of electrons, and so energy sources are sometimes called electron donors. In the case of glucose metabolism, the energy source is glucose. We can use other sugars, we can also use lipids and proteins as energy sources, but we're going to be starting with glucose, and then we'll talk about how we can use some other things in there as well. Sorry, on the same slide. The terminal electron acceptor is what's eventually going to accept those electrons. Again, we're going to be passing those electrons from one molecule to the next in a series of redox reactions, but the energy source is what we initially get the electrons from, and the terminal electron acceptor is the final element that is going to receive those electrons from the electron transport chain. And again, all aerobic organisms use oxygen as the terminal electron acceptor. Redox reactions are just reactions that involve the transfer of electrons from one molecule to the next. Anything that gets oxidized is going to lose an electron, so the glucose is going to be oxidized during glucose metabolism. Anything that is reduced is going to gain an electron, so our electron carriers they're going to be reduced at first. So the glucose, it's actually a submolecule, but another molecule of carbohydrate is going to give an electron to like NADH. So NADH will be reduced in that stage. But then the NADH is going to go to the electron transport chain and give the electrons off, so it's going to be oxidized at that point. So it's all about transfers of electrons, and so where those electrons are going helps tell you whether something's going to be oxidized or reduced. There is a mnemonic that I gave you in the notes to help you remember what's being oxidized and what's being reduced. It's called oil rig. What that stands for is oil is oxidized is lose an electron, and then rig, reduced, is gain an electron. So that can help you remember what's what. I won't do that one. Next up, explain the role of the electron carriers and mention the three listed in the book, include the oxidized form and the reduced form of each carrier. So what I did in my notes is created a little table, and on one side of the table I have oxidized, and on the other side I have the reduced form. So I'm going to start with the oxidized form. You have NAD+, that's actually this molecule, it's nicotinamide adenine diphosphate. NAD+ is the oxidized form. When we reduce it, it becomes NADH. Next, FAD+ is the oxidized form of FADH2, and then NADP+, which is this one up over here, that becomes NADPH. Now, NAD+ and FAD+, those are going to be helping with proton motive force. Those are the electron carriers that give the electrons to the electron transport chain. NADP+, though, that gets used in biosynthesis. Principally, when we talk about that, it's going to be in photosynthesis, and I only talk about photosynthesis with the majors. I don't make the non-majors do that, because nurses don't tend to need to know a lot about photosynthesis. So this is kind of what I was giving you in the notes. So the oxidized forms, NAD+, FAD+, NADP+. The reduced form, NADH, FADH2, NADPH, and then what are they used for? I said all of those guys off over there. Then for explain the role of these, they're just electron carriers. They pick an electron up from one molecule, they drop it off at another molecule. So they're just shuttling electrons around inside the cell. Next, what are precursor metabolites, and provide some examples. They're intermediate molecules that we make in a pathway that can be shunted off and used for something else present inside of a cell. One of the first examples we're going to talk about is pyruvate. Pyruvate can either continue through glucose metabolism by entering the transition step, or we can siphon it off to make some of our amino acids like alanine or leucine or valine. And so it's a molecule that can either continue through the cycle, or we can use it for something else present inside the cell. There's going to be, I think, 6 precursor metabolites in glycolysis, and I think 4 precursor metabolites in the Krebs cycle, and we'll talk about those when we get to those. Hey, look at there. There's some of our precursor metabolites that can be shunted off. The example I gave you was pyruvate, but there's others that I didn't go over, but these are all examples so far. Alright, so your book uses the analogy that a metabolic process is like a highway. Explain what that means and use glucose as an example. Okay, and I'm going to use I-35 as an example. So I-35, the interstate that runs through Alvarado and Fort Worth, Dallas, that goes all the way up to Minnesota. You could be like glucose and go all the way through that process and end up in Minnesota as carbon dioxide and water. Or you can take off-ramps and go visit something in Missouri to see what's going on in Missouri and then go off to Washington, D.C. You can take off-ramps. If a molecule takes an off-ramp, that means it's a precursor metabolite, and that means there's another reaction that your cells or the bacterial cells think is more important than generating ATP at that point, and so we'll siphon it off and we'll go do that other reaction. So anything that can take an off-ramp is a precursor metabolite. One of the beefs that I've always had with textbooks is they will tell you that you can generate X number of ATP in this process. That does not ever take into account that your cell can work like that highway system and siphon things off to use them for other things. So the numbers that textbooks give you are always a theoretical maximum. That means if you were to take glucose and burn it completely, you could get, in theory, up to like 34-ish ATPs. But in reality, some stuff decided to take that off-ramp, and if they take the off-ramp, you can't make ATP out of it because it went to go do something else inside the cell that didn't have anything to do with ATP metabolism. And so that's how it's like a highway, is things can either stay on it and go from point A to point B, or they can go off and go visit point C before they get to point B. Okay, that's where I'm going to stop this video, because next we're going to start getting into a little bit more detail on glucose metabolism, and I wanted to kind of give my voice a rest before we get there because the process is complicated. So there will be a second video for this chapter.